I'm Zhao and currently a tenure-track associate professor at Shanghai Jiao Tong University. And it's really great honor to be here and to give this talk at Sydney Precision Data Science Center. And today my presentation is to share our work named INSPIRE, which is for interpretable, flexible, and spatially aware integration of multiple spatial transcriptomics sections. So I think as we're in the field of statistical genomics, we have all seen the great success of the single-cell genomics, where the cells are really the basic building block of our life. And with the single-cell technology, we can measure the gene expression from each individual cells. And recently, there are newer technologies named spatial transcriptomics or spatially resolved transcriptomics. So compared to the single-cell RNA sequencing or other modalities, basically they capture the molecular information from each individual cells. But what is new about the spatial transcriptomics? It brings another layer of information, which is the spatial context into the data set. Now we can not only see the spatial, the gene expression on each location or each spot or cell, we can also know its spatial locations. So the field has been really active. There are a lot of different technologies and platforms developed for these type of the data. So given the limited time, I will not introduce each of them, but for simplicity, we will basically regard the data type as two matrices. One of them is quite similar to the single cell data. For each row is a spot or cell and capture all of the information from different genes. And for each column, it measures the one particular gene, the value of its expression across different spot or cells. And very importantly, we usually say that the spatial transcriptomics bring the spatial context information. And in terms of the data format, it besides the gene expression matrix, it also provides the spatial location information paired with the gene expression. And more specifically, if we focus on one of the spot or cell, which is basically one row, then there are spatially the XY coordinate information here associated with certain spot or cells. So basically, every data set contains those two types of the matrices. And there are lots of publications, and more importantly, there are richer and richer data set accumulated for the field. So there are not only the number accumulated, but also the species and also the organ or tissue types, and even across different biological conditions. So given those rich data, so of course we want to ask what type of the biological questions we can ask from those spatial transcriptomics data. I think as I started the talk with that, the ST data offers the spatial context besides the single cell data set. So in my opinion, so I think if we want to focus on the analyzing the ST data, we should focus on the spatial context. So basically during the postdoc training, I have been very interested in two of the aims. One of them is to understand the spatial organization of the tissues or organs using the ST data. And more importantly, I want to combine different type of the sections together to distinguish are there any shared signals or are there any signals specific to certain biological context. And another aim is uncovering the cell-cell communication in tissues. And recently, we have a preprint out for the second aim, but today the presentation will only focus on the first one. So talking about uncovering the spatial organizations of the tissues. So recall that we have those high dimensional gene expression measurements. So if we focus on each gene, then we have the expression across spot or cells, and we have the spatial location information associated with it. So you can imagine there are 3 columns, and they can form one particular image. So you can imagine this section form an image, but only for one gene. And if you have 1,000 of gene, then you have 1,000 or hundreds of such image. So we want to analyze such data and to answer that, are there certain group of genes tends to co-localize together to form certain functions on those tissue. And we also want to know, are there any spatial distributions of them on the tissue? So to answer that question, actually there are already existing methods. So we want to understand, are there gene modules? So the gene modules are basically the group of genes usually co-localized together spatially. And so for understanding the gene modules and their spatial distributions, there are existing methods. And without going into too much details here, I want to summarize that the pioneering studies will basically decompose those high dimensional data into the low rank approximations. And very importantly, they use non-negative matrix factorizations, in short NMF, which means that all those values in those low rank approximations are non-negative. So you can imagine there are high dimensional data sets, and the true dimensionality is basically not as high as the original data set. And those methods aim to capture the true dimensionality, but also achieve the interpretability with those non-negative values. So in this page, I want to illustrate why those methods can achieve interpretability with the non-negative values or with the NMF. So there are methods coming up in nature genetics and nature methods, all in this topic. So imagine we have already decomposed those one data set into the low rank approximation. So we name those cell or spotlight factor matrix as the spatial factor matrix. And here we also have a factor by gene matrix. We name it as a gene loading matrix. So we'll zoom in one by one to see why it achieved interpretability. So if we focus on one of the factor, then there are a lot of non-negative values. Each one corresponds to one gene. So recall that they are non-negative, so we can basically learn the score. So the higher score means the higher importance. So given one factor, we can directly see that what gene is really important to give a very high weight on the factor. So with that, given a certain threshold, we may know, okay, there are two genes or 5 genes that are really important to define that factor. And those group of genes will be named as gene modules in the field. So now knowing the meaning of the gene, the loading of the factors, now we can see one of particular factors in the factor matrix. So still recall that those are still non-negative values. And we can map it with the spatial coordinates to map the factor value back to the whole section. Then what we can see is the spatial enrichment of that factor. So suppose we now know there are 5 genes form a gene module for the factor, then we can use the geo analysis to know its biological meanings. And with the factor matrix, now we can see the spatial enrichment pattern on that. And you can also imagine if we combine multiple factors together, then we can really uncover the very fine-grained organization of the patterns out of the very high dimensional and hard to analyze data set. So this slide, I want to use that to show why we care about the NMF and why it achieves certain interpretability. And this page shows that, so the previous page shows that what happened if we focus on one of the factor. And this page shows that you can imagine given a data set and with those decomposition, what you get is those mega genes or gene programs with those meaningful genes. And also if we combine multiple different factors, you can really uncover the fine-grained organization of tissues like here and here. So those methods are really good and state of the art in the field, but one potential limitation is those methods are primarily designed for one section at a time. So as shown here, at one time, it comes a cell by gene matrix and also the spatial coordination. But so going into those methods, it can only handle one section at a time. Then you can imagine that there are different use cases like shown in this figure. Like if you want to get a comprehensive understanding of a very complex organ or tissues, basically you may need to focus on one axis and get parallel sections along that section, along that axis to get multiple sections. Or maybe you also need to change the views to get the sections from different angles of the tissue so that you can ensemble them together to form a very comprehensive atlas. But the problem is the previous method, like the NMF method, handles one section at a time. Then suppose one method handles this one and this one, each report like 30 factors. Then you really don't know the matching between them to how to align them. And also it's really hard to know, are there any specific factors? And more importantly, the factors can also be confounded by the heterogeneous unwanted variation. So here there are many different use cases, but I use two of them to let you know that the previous method is really good at handle one section at a time, but it's really not enough for the comprehensive applications. So besides ensembling the whole atlas, so now in the field has some researchers are really interested in learning the developmental dynamics. So it will ask, so that type of study will really need not only one section, but instead different sections, maybe one or multiple sections at each continuous time point. So I use this figure to show that if you focus on those type of biological questions, you do need multiple sections to jointly analyze together. So to summarize, what is the limitation and challenge of current interpretable method? So the previous method only handle one section at a time, but now we need to uncover both like shared and context-specific gene programs together, like simultaneously for multiple sections. And what is the challenge? The challenge is that suppose we have 10 sections. There are technical variations and there are like batch variations. So there are really complex unwanted variation hidden in those data sets. So we want to distinguish shared signals out of those noisy and unwanted variation. And it's really challenging because some of the sections may even capture the fine-grained but context-specific signals. So there are three sources of variation. Unwanted variation as number one, and number two is the shared signals, and the number three is the biological meaningful but maybe section-specific signals. So we do need methods to distinguish those three sources of variations in order to achieve the interpretable analysis for multiple sections. So here, we want to introduce our method named INSPIRE, which published in Nature Genetics in this year. So it combines the deep learning thinking and also with the interpretability from the traditional statistical modeling. So later on, we will see how can we integrate those two components together to achieve the flexibility and also the interpretability. So here, to introduce our method, I want to start with a single section modeling, because if we understand that, then it's quite natural to extend it from one section to multiple sections. So start from one section modeling. Recall that for each section, we have two matrices. One is the gene expression matrices. Here we use the big Y to denote it, and also the spatial location information. So here, because every cell is located spatially and within certain microenvironment or niches, so here, to handle those type of data, it's really important that at each time we focus on the center cell. We not only want to analyze it independently, but instead we want to understand the communication or importance score from other neighboring cells to it. So here we use the graph attention neural network to connect every cell with its neighboring cells and to borrow information across different cells with the adaptively learned weights. So with that, we embed those high dimensional data into a low dimensional representations. So here in the latent space, we have latent embeddings. So I think it's a quite mature technology for not only the spatial transcriptomics, but also the single cell data set. So we want to have the latent representations out of the high dimensional data. And importantly, how can we achieve the spatially aware modeling together with the interpretability? Here we use a linear neural network to generate those beta, which is the spatial factor matrix with non-negative values out of the Z. So here we use a neural network to generate and then combined with a non-negative loading matrix. So recall that to achieve the interpretability, we need the gene loading matrices to describe the signature or the gene modules and associate with the beta to learn the spatial distributions. So together, those beta and mu form a low-rank approximations to approximate the high-dimensional data. And also, this is the idea coming from the previous work I have just briefly introduced. So because the whole idea is to use the low-rank approximation to approximate the data. And of course, the loss function is coming from those, but we also need to account for the count data, the count nature of the data. So we use the Poisson distribution at the last layer to form a good approximator. So without going into detail, in this page, I really want to show that we embed the structure into this latent space. And then coming out of the latent space, we want to achieve the interpretability through the NMF and together approximate the original data. So you can imagine the whole algorithm form a kind of system from this input and also its approximation. So I use these slides to show that also to remind you like how can we achieve the interpretability. So imagine, suppose we have those sections. Then in the latent space, what we can achieve is we want to preserve the structure from the representation learning. So suppose there are trajectories hidden in the data, then ideally we may need to look at the latent representations to call out the structure. And also, the gene loadings and factors achieve the interpretability. Like here, like I previously talked about, we can get the importance score to form gene modules and also visually look at its distribution on the tissue. So those three components each have different functions. And now after introducing how can we achieve that, basically reformulate the previous method with the deep learning. So here in this page, we show that how can we adapt those traditional methods into the deep learning framework, but still achieve the interpretability in the single section modeling. And later on, here I want to show that now how can we achieve the analysis of the multiple sections simultaneously? So of course, we may all think that it's very, very natural to extend it to multiple sections. We only need to change the input. When we develop the method, we share the same feeling. So we start with the multiple input. So here we have 3 sections. Oh, here is 4. So let's assume we have 4 sections. So we have 4 big matrices for gene expression. And of course, for each section, we can also build a graph. We can also build a spatial graph. So then naturally, we think that still we use the shared graph attention network. We can, of course, still map the high dimensional data into the latent space. And then suppose we preserve the good structure of the data here in the latent space. Of course, we can also still use the linear neural network to generate the non-negative values for each of the section. So we have 3 to 4 sections, say, for those spatial factor matrices. And to achieve the interpretability simultaneously for the multiple sections, very importantly, here we should keep only one gene loading matrix, because we want to understand that for one particular factors that we know its meaning and is shared across sections, and we want to know what are specific distributions of it on different sections, which means that if we want to achieve the interpretability for multiple section analysis, it's very important for us to have a shared gene loading matrix. And then combine these one shared gene loadings with multiple factors matrix, one for each dataset. We can, of course, then approximate the original dataset with those 1 and 3 factors. So it's a very natural extension from one section to multiple sections, but later we will show that we do need additional designs to achieve the data integration and interpretability to extend from one to multiple sections. Without going into detail here in this page, I want to illustrate the big idea. So here, basically, we want to achieve the integration in cell level and gene levels. First of all, because the dataset carry unwanted variation, you can imagine using a shared neural network to map it from the high dimensional data into the low dimensional embeddings. The low dimensional representation may affect it or confounded by the unwanted variation. So here in the cell level alignment, we want to correct the representations of cells or spots in this latent space to achieve the harmonized representation. Okay, so now we assume we have certain designs to achieve the harmonized representation. And then we can think that now the latent embedding is free from unwanted variation. And with the shared neural network, we can also assume that, okay, now the spatial factors, they may be different across different sections, but it's also free from unwanted variation because it really generated from a very clean sources. And then times the shared embeddings, now the problem is the raw expression is very noisy and have certain confounding factors. And we have really clean factor and shared gene loadings. So I think now we understand that we need additional designs to bring back the unwanted variation into those approximations so that it will not be confounded by that. So that comes from that lead us to the gene level correction. So later on, I will go into the detail of them, but here I will go all over the big idea. And later on, we have those 3 components extending from one to multiple section. So though here we have the embeddings to see the structure, and here we have the shared interpretable gene modules. And if we project it onto different sections, then we can analyze the enrichment of different pathways one by one or jointly across different sections. So I hope I made it clear how we basically want to design our method. And here I want to go a little bit into the detail of the different like cell level integration or gene level integration. Here, we focus on the cell level integration first. So coming from the high dimensional data and going through an embedding encoder, we want to encode information into the latent representations. So then you can imagine that each one of the sections form a data distribution. Let's suppose we have section 1, 2, 3 until the big S. Then the strategy we want to introduce is we focus on one pair at a time because it's really difficult to correct for multiple sections at the same time. So now the strategy is suppose we focus on the section S and section S plus 1. So it's two adjacent sections and each form a data distribution in those in those latent embeddings. So here we make use of the generative adversarial training. So still without going into too much detail here, I want to introduce the basic idea. So given those two samples from two distributions, we want to align those distributions together. The strategy we use is we introduce a discriminator to tell the differences between them. And those signals reversely supervise the encoders to mix those two distributions together. So without going into too much detail, you can imagine that we introduce some additional neural network to help to get the distribution matching between each pair of the sections. So suppose we have section 1, 2, 3, 4, then we need to introduce 3 additional discriminator components. One for align section 1 to 2, one for 2 to 3, and another for 3 to 4. So with those design, we can align the embedding very well, like this one. And also, like I said, there are additional sources of variation which captures the biological meaningful and dataset specific one. So which means maybe there are certain components in the distribution that shouldn't be aligned so perfectly across different sections. So here, given the time, I will not go into detail, but also briefly talk about there is a very important design in our framework. So there is a design for the discriminator, and intuitively, if two distributions are different, are really easy to distinguish from each other, meaning that there are maybe one component that really distinguish from others, then the discriminator will overlook it, meaning that we will only supervise the mixing of those shared components, but will leave the additional specific population as it is. So here, basically, here is how can we achieve the cell level alignment, and we should also keep in mind that we have a specific design to capture the specific signals. And also, we still go back to this figure to see that now it really integrates together and free of unwanted variation. And now we move to the interpretability component to see how can we achieve that. So here, empirically, we find that we need another layer of the gene level correction. So this is clean out of the free from the unwanted variation, and this is shared and of course free from unwanted variation. So we need to add the unwanted source of variation back into the NMF so that we can approximate the raw data. So here we introduce the section and gene specific effect back into this noisy source, back into the clean NMF so that it guides the training to be well learned. And also the last component for our model design is we want to preserve the meaningful structure in the latent space. So to do that, we use the geometric consistency. It's very easy to understand, and the intuition is that we can learn the similarity matrix from each pair of the cell or spot. And if it is very similar, then the similarity should be preserved in the latent space. And if it's less similar, then that differences should also be preserved in the latent space. So you can imagine using the raw data, we can calculate one cosine similarity matrix. And using the latent embedding, we can also calculate the cosine similarity matrix among each pair of the cell or spot. So very intuitively, we want to compare those two similarity matrix and to let it to be very similar so that any structures, especially for the trajectories, should be very well preserved in the latent embedding. With all those components together, we will build it into a unified framework. So it's not a step-by-step method. Instead, it's really a unified optimization problem. And after training, we have three sources of output. One is the latent embedding, one is the vector, and one is gene loadings. And here I will use this figure to show that they really have different meanings. So if you want to look at the rough structure or the very broad component of the section, you may want to look at the latent embeddings to see its structures. And also, if we want to achieve interpretability, of course, you may want to look at the shared gene loadings to understand the gene modules and biological pathways if we also conduct the enrichment analysis. And knowing each meaning of each factor, we can then look at how to compare it across different sections. Like, are they basically connected across different sections? Are those very unique to one section? So we can look at the very fine-grained organizations of the meaningful factors using the spatial factor matrix. Okay, so now we move to the result section. So I will very briefly go over what we can achieve using this method. So of course, as an initial step, we focus on the benchmarking dataset in the field, which is this DLPFC dataset, and we show that after jointly modeling multiple sections, we achieve better scores quantitatively for uncovering the structure, because we can really borrow information across different sections in not only the latent embeddings, but also the spatial factors. So those are very important components to borrow information across sections. So you may know that the SPACE-mix and NSFH, which conduct NMF for one section, it also perform well, but it's less. So our method can achieve better results because we borrow information across sections. And also, importantly, we want to show that not only we can achieve better representations in the latent space, very importantly, we can recover the factor and loadings accurately. So here we use a simulation study to pre-design some factors and gene loadings, and to show that to compare the performance among different methods for recovering them. And also, you may see that the NMF-based method perform very well compared to other categories, but also within the NMF, our method achieve better results. And also, we focus on, so it's also a kind of a validation experiment. So we collect sections from mouse brain with known structure. So we have studied those mouse brain very well. So we collect two from the sagittal and one from coronal view. So they capture different view and of course different structures. So using our method, we can recover the organization very well and also preserve the trajectory and also capture the specific signals. But other method, if you compare the spatial-mix, it cannot integrate data very well as it's designed for one section at a time. And also very famous method like precast. So maybe I can talk a little bit more for that. So it assumed that the latent space is coming from a mixture of Gaussian. In that sense, it really decomposed the continuous trajectory into different clusters, different Gaussian distribution components by the nature of its design. So it basically cannot preserve the structure very well. And also here, it's also affected by the unwanted variation. And also, if we look at the factors and loadings, of course, we can show there are meaningful loadings. We can validate those markers by the literature and also to see its organizations. So given the limited time, I will quickly go through the applications. So also, if we focus on Xenium data, which is a very high resolution and newer platforms, and here we analyze a brain cancer section containing 3 different neuron subtypes, and one is in color green is more invasive, and 2 is less invasive. So although we carefully examine different factors, they have different meanings. But here, we only show some of them here. Like, for example, interestingly, we find that there are 3 distinguished factors. Like if we use enrichment analysis or analyze these markers, they all correspond to the myoepithelium. So interestingly, they show different patterns of gene signatures. They also show different colocalization patterns in the spatially. So at one time, we conduct the enrichment of the colocalization for one factor among those 3 types of the tumor subtype. Then we find that interestingly, like the myoepithelial 1 only tends to be colocalized with the one of the particular subtype that is less invasive. And instead, like the myoepithelial 3 tends to be jointly colocalized with 2 of those neurons, tumor subtype. And also, likewise, we find 2 endothelial cell population. And very interestingly, one of them tends to colocalize with the invasive tumor type, but the other don't show such pattern. So it helps us to understand very detailed, like fine-grained patterns within the tumor microenvironment. And here we also apply our method to the understand the developmental trajectories. So here we collect those Stereo-seq data set. Like if I remember correctly, it basically covers like 500,000 spots. And here we can know that how the organs or tissues grow bigger and bigger with different proportions along these trajectories. And also we find out some of the organizations do not show in the previous stage, but later become clearly visually seen in later stage. And also, very similarly, we can understand the more fine-grained structures with its biological functions by the understanding its gene signatures and also the spatial locations. So so at last, I want to use like maybe 1 to 2 minutes to talk about other applications. So previously, we were talking about the representation learnings and also the interpretability for analyzing multiple sections. And here, we want to show that our method, the interpretable method, not only achieved an interpretability, but also like the interpretable representation is really good for other downstream analysis. So here, like shown in this figure, so there are different parallel sections. So the the whole aim for the study is to reconstruct the 3D structure of the tissue and to understand it in the 3D world. So there are different methods. They are all very good, like Morse code and cast for the non-rigid transformation physically to align those sections. And without going into the detail, but their idea is they either use like principal component analysis as representation or using the embeddings from like SCVI or their own neural network. They make use of the embeddings and build upon that to make some physical alignment across different sections. And here in this page, we want to show that our method, if we plug in our learned interpretable representations inside into the other like well-established method, and instead of using PCs or embeddings from SCVS, if we use our learned interpretable representations, we can help the other method achieve better results. So here, like the Inspire plug-in into the Morse code, it achieves even better result than the Morse code. So in summary, here we introduce our method and for data integration, and we achieve the interpretability through the joint NMF. And our method is really scalable to handle millions or cell or spot. And very welcome to check our paper in the Nature Genetics and try our software. And lastly, I want to acknowledge the help and guidance from Yale. So I only want to thank my mentors, Professor Hongyu Zhao and Professor Wei Chang from the School of Public Health and School of Medicine. And I want to acknowledge great collaborators who really contribute a lot to this project. And especially in Xinlin from this center, we are really good collaborators, and she helped a lot in this project.